Okay.
Okay. I think we can probably go ahead and get started on the webinar today. Thanks everyone for joining. I saw it's recorded. If you're interested in more Beyond the Scope talks, these have been recorded and they're on our website. I'll post a link a little bit later. But with these, we like them to be fairly interactive as well. So ask lots of questions. We'll have time for that at the end. And so with today's talk, as Tong and I were talking about a little bit, I'm excited to hear more about kind of the light microscopy facilities that we have on campus here. CMIF is nice and complementary to a lot of things that we do here at CMAS. So we've actually worked with them quite a bit with sample prep and other techniques. So to give our talk today on different light microscopy techniques, we have Tong Zhang. He's the Associate Director of CMIF, Senior Microscopist, and he does basically all a light microscopy there. So I'm really excited to hear about your talk today.
Okay. Okay, I'll start and I'll stop sharing my video and then I'll just talk so you can still hear me, have a better quality for the videos. Okay. Let me see how can I do it. So stop the video. Great.
Yep, yeah. I can hear you.
Okay, great. Okay. Make sure you can still hear me. Okay. First of all, thank you for the invitation and allow me to give a talk to the OSU CMAS audience. Since many of you are expert on electron microscopy, I'd like to use the opportunity to talk more about the light microscopy. So which is also the major service at the CMIF. So I wrote on the title is Novel Light Microscopy Technologies, but many concepts actually were developed a long time ago, and only recent years have been successfully developed and commercialized and widely available to the to all the researchers. Okay, so you can see the screen here. Let's see. Okay, so now we all know microscope has become an icon of science. So you see this on TV. So for example, the few years ago, the most popular one is the Sherlock, and you can see he's looking at one of the a microscope looking for some samples, and also you saw it in news. For example, when Obama was the president with the NIH, and those scientists showed him the stem cells, and he looked at one of the microscope to look at those cells. And also you saw it on you probably saw this on your neighbor's $100 polymer banknote. So this is kind of the $100. So on this on the back this $100, you can see there's a lady looking through a microscope. So if you haven't seen enough type of microscope, you will recognize. So this is a Zeiss actual imager 2 microscope. Look like this one for commercialized. Okay, and just in case you're curious about this person, the height person here, and he is not the person who found out that the insulin, and he actually he's the Robert Borden, the 8th Prime Minister of Canada, in office in 1911 to 1920. So for a microscope, actually, is mainly made by 3 parts. First of all, the detector. It could be your eye, our camera, or PMT for confocal, and also the magnifying lens. And on the magnifying lens, usually we're talking about the objective. You have the magnification, numerical aperture, and also corrections. And those are very important for the property of objective. And also you have the illumination source, for example, the halogen for bright field in the back in this green circle. And also the mercury, for example, or the LED become more popular right now, or the XSAT. And also the lasers for confocal system. So when you look at an objective, you see there are lots of information actually labeled on an objective. And you can see, for example, they're like tell you what type of objective it is, and the magnification, the numerical aperture, and what kind of property the objective can handle. For example, in this case, DIC emerging, there's correction color. And they're also working for different thickness glass, for example, in this case, range from 0.19 to 0.15 millimeter. And there's one widely used called the number 1/2 cover slip. Actually, it's 0.17 millimeter thickness. And it doesn't matter what brand of microscope you use, all the objectives calibrate for the 0.17 millimeter of 1/2 cover slip. That's become a standard. So in this case, in this picture, there's a correction color corrected for the water, W here, all glycerin for different immersion. And also, people don't notice this anymore because sometimes those color ring will be wear out. And also, usually when people work on a microscope, they turn off the light so they don't see the color on the objective. So the color actually there's meanings. So for example, like they can be a standard one or polarized one for the DIC, or even for the phase one, you use different colors. And even for different magnification, they can use for different colors. And also for different immersion, also use different colors, but at different position. Okay, usually we are talking about the final magnification is the magnification, the ocular times the magnification, the objective. So usually the magnification for ocular is 10x. So in this case, it's a 63x. So 10 times 63. So the final magnification is 630 times. Unless you use other magnifying lens in the light path. So that's your final magnification. And if you cut through an objective, you're going to find out there's a series lens in it. And why it's so complicated is because it's very necessary due to the property of light. So when the light passes through a convex lens, even the same color of light will focus at the different focus due to the curve shape and also different thickness the lens. And this phenomenon is called a spherical aberration. So you can see in this picture here, when the light passes through different parts, the lens will focus at different focus. And for different wavelengths or color of light, when they pass through a convex lens, different color will also focus at a different focus. And this phenomenon is called a chromatic aberration, which is shown in the middle picture here. So for example, the blue light has a higher frequency, so it's bent more. And so the focus is closer to the lens than the red one, which has a longer wavelength. And also when the light passes through a different angle lens, a different color also focus at different plane here. So this phenomenon is called a field curvature. So a good objective should have correction for all above. So and also become more complicated and also expensive. And here's just the table for different for common correction lens name and the meanings. You can see, for example, the best one usually you see is called the plan apochromatic. It corrects for multiple colors and also corrects for the field curvature. So therefore, give you a flight image. And the optical resolution, or called the diffraction limit, was first theoretically given in mathematical formula by Ernst Arby in 1873. So at the University of Jena in Germany, there's a monument. So on the monument, there's a formula you can see here and also showing here. The D is for the resolution. The smaller the D value, the better the resolution. And the lambda is for the wavelength. And the N is for the refract index. And the sin alpha, the alpha is the angle of the focus, the half the angle of the focus light cone. And usually people think this picture is Carl Zeiss, but actually this is Arby picture. And this is the Carl Zeiss. And together with another person, Otto Schott, they worked, that person working on the glass. So they together established a very successful optical instrument company, Zeiss, until today. So this formula defines any optical instrument resolution. So you can see from the middle one here, there's a one property for any optical device is called the numerical aperture. So in this case, for the objective here, the numerical aperture equal to the refract index times the half the angle the focused light cone here. Okay, so for regular for the common immersion we use for microscopy, usually air is 1, water is 1.3, and glycerin is 1.5 almost. Oil is more than 1.5, and there is special oil is with an even higher refract index. So based on this formula, the higher the refract index, the smaller the D value, the better the resolution. So therefore, the larger the NA value, the better the resolution. So in this case, we see here there's a 63x, that's the magnification, and with a slash 1.4 oil, that's the immersion oil, 1.4, that's the numerical aperture. So when you put in this form, and also in this this picture here, you can see. So for example, usually in the A, that's probably the case for an air objective, which is with a longer working distance. But for the half angle, the focus cone, you can see it's much smaller comparing with immersion lens here, which is much larger angle here. And also based on the distance, you can see the NA value become larger with the increase of the alpha value here. Okay. And for visible spectrum, usually we're talking about from 400 to 700 nanometer wavelengths. So based on the obvious diffraction limit equation calculation, a microscope has a resolution limit about the 200 nanometer. That's the UV, as you can see, is about 0.21 micrometer. So there's the diffraction limit. And since 1873, we discovered any optical device, and no one can go beyond 200 nanometers. And study life science with light microscopy, there are many advantages. So for example, it can specify labeling for certain protein or RNA or even DNA, and can also do live cell imaging, provide spatial and temporal information about a biological process. And it's easier to do quantitative analysis. Or those are missing in electron microscopy. So due to the diffraction limit of light, so for less than 200 resolution, so right now we so-called the super resolution. And usually, for example, for virus or protein or even small molecules, those are way below the optical resolutions. And here's the famous imaging challenge, so-called the internal triangle. So in microscopy, also kind of like live, similar to live. So optimize in one capability will come with trade-off of the other two. So for example, resolution, speed, and then sensitivity all can be also called the signal to noise ratio here. So for example, you can image faster speed. However, you will sacrifice with the resolution and then the sensitivity. So for any good image, it should have enough information. So the information we're talking about, first of all, is magnification, which is we have the objective with different magnification, which is usually how large you can see this image, right? For example, if it's too small, you can't see very well. And the second one is the resolution. If the resolution is too low, then you're not going to see any detail on this picture. Even now you can see this picture larger than previous one, but the resolution is low, and then therefore it looks really blurry. And another one is called a contrast. So it's kind of like a signal to noise ratio. So if the signal to noise, in this case, you can see there's like a fog across this picture. So therefore this picture, you don't see the detail very well. It's just not enough contrast here and also signal to noise here. So altogether, reach to the optimal will give you so-called a good image here. Okay. So before talking about the super resolution, I will introduce there are other really clever ideas to get a so-called good image. So first of all, I think many people are familiar with a term called the deconvolution. So this actually term was first introduced to life science in 1984 by David Agard. And I think this guy is also very famous in the electron microscopy field, and also John Sidat at the UCSF. I wish to mention John Sidat's wife, Elizabeth Blackburn, and also won the Nobel. That's his wife, and won Nobel Prize in 2009 in medicine and physiology for the study of telomeres and then telomerase. So he first introduced this term to life science to solve the problem for wide field fluorescent Z-stack imaging, because at that time in 1984, the confocal is not very popular. So what is deconvolution? So for example, in this case, we have a subject here, and also a so-called point spur function. The point spur function is assume optical, everything is perfect. And it's a mathematical function so that it describes the distortion of light went through this system. And together they give you an image. So after we have this image, if we can find out this PSF, the point spur function, so from the objective times this point spur function, get this image, this process called a convolution. And the reverse operation is called deconvolution. So we have an image right now. If we know the point spur function, we can calculate this, and this subject with a much more sharper image and undistorted image. So this idea actually is not new, and it's already being used by astronomers for clearing their images from telescope. So for example, usually the observatory are built on the high altitude mountain to avoid far from the city, to avoid the light pollution, and also the most important one from the atmosphere turbulence. So in this case, for example, there's a nebula picture here. The left one showing you is the before deconvolution, and the right one showing you after deconvolution. You can get a much sharper image. And the same application applies for the biology. You can see this a dividing cell. I think it's in the anaphase. You can see the blue in chromosome, and all those dots are kinetochores. So after deconvolution, you can get a much sharper image. So on this top picture, you can see one of the observatory shoot off one of the a yellow laser point to something. So I was curious what they're shooting at. So actually there is so-called optics. So this term actually was first introduced by Horace Babcock in 1953. At that time, it's like a science fiction. So actually this idea was also written in some science fiction novels. So the idea is, because the atmosphere turbulence, so therefore this image will look like a really blurry. So if we can have some like a magic device, in this case, for example, called a deformed mirror, can correct for this thing, then we can restore this image, become a sharper one. Okay, this idea is very clever, but until in 1990s, it's become possible thanks to the development of technology, the manufacture, and also the advanced computing technologies. So this laser, this yellow laser you see actually is a sodium laser using as a guide stars for this adaptive optics for the correction. So when they use this laser as a guide star and then use a deformed mirror correct for this distortion, and therefore you get a much sharper image. So this is different than the deconvolution. Okay, just based on this adaptive optics technology. So for example, at the bottom, this is like from the observatory. So for example, this is the first one you see before the adaptive optics. It's a really blurry image. You cannot see anything. And of course, that's why the scientists sent out the Hubble Space Telescope to space. There's no atmosphere there. So you can get a much sharper image in this case here. But with adaptive optics, even the ground observatory can get a very sharp image, can compete with Hubble telescope. In this case, you can see the Neptune, one of the planet, in a very sharp image, very similar to the Hubble Space Telescope. This is quite an amazing achievement by just using this deformed mirror. It's a little bit similar to right now. It's so popular for the called noise canceling headset, right? You can have another object phase of sound cancel the noise. This is kind of a similar technology, but it's correct for the light distortion. So similar idea was used in life science back to 2010. So by Eric Benzik, and then Naji is also his wife. They work together. So one of the a similar situation they saw in biological sample is when light pass through this biological sample, it's also being distorted by different part of the sample. So which is similar to the telescope situation, you have the atmosphere turbulence here. So by using, for example, either the optics deform the mirror in here, or using wavefront sensor to correct it for this, you can get a much sharper image, especially in the inside the animals or in the sick tissues. So this is without the adoptive optics, and this is with the adoptive optics. You can see it's a much sharper image. Okay, so we have been talking about the two cases which is borrowed the idea from astronomy. One is the deconvolution, and the other one is the adoptive optics. And then the become most popular one is the confocal, where first the concept was introduced by Marvin Minsky in 1953. He patented it. And you can see this is the original patent drawing, which is actually very similar to the current design. For example, in this case, like this, in this case here, showing on the right. And back then, there's no laser because the first laser was invented in 1960s. So that's 1953. So you can see actually from the drawing is using, I believe it's like some very powerful mercury lamp here. So but right now it's using laser. But the magic of confocal is not the laser, actually it's the pinhole here, okay? So the pinhole will only allow the light in focus go to the detector, give you a sharp image. And any light out of focus will be blocked by this pinhole here. So therefore this pinhole will change based on different wavelengths and also refract index and also the situation. So, and also on any confocal system, there's one unit labeled the pinhole is so-called the Airy unit. So we know equal or smaller than one Airy unit that's confocal, larger than one is not confocal anymore. They give you more blurry image. If you fully open the pinhole, it's like a Widefield image. And people also know that if you close down the pinhole, if you get a much sharper image, and that means also a higher resolution image. But the problem is if you close down the pinhole, there's less light go to the detector. So therefore you suffer this back to the internal triangle problem. You have much lower signal to noise ratio. You get much noise image. And before I talking more about how people resolve this problem, so let me introduce a so-called Airy Disk. This is also introduced back to a long time ago, 1835, by George Airy. He was the astronomer Royal, and he was the first person theoretically calculated this Airy Disk, which is the size of the Airy Disk depend on the wavelength and also the size of the aperture. So for example, in this case, you can see in this case, the center, when the light passes through a small pinhole, if everything is optimal or aperture, for example, if you form this ring pattern, so because of the diffraction property of the light, so in the center will become a very bright spot and then become dimmer and form another brighter ring and dimmer again and another ring. So the center spot is called a one Airy unit. And also you can see when two subjects are getting closer to each other, in this case, we can still resolve. So that means the resolution can still separate the two subjects. So in this case, it will appear as one, just brighter in intensity. And Zeiss is cleverly invented something called the Airyscan detector. They announced this technology in 2014. So instead of using one detector, they use a detector array. So each small element is one detector and equal to 0.2 every unit. And the diagonal direction is a 6 detector, so totally it's about 1.2 or 1.25 every unit. And together they give you much higher signal to noise ratio. And summarize all the point spur function with 32 images. They can reconstruct an image with a much higher resolution than conventional confocal. So right now they advertise the XY resolution is about 120 nanometer and Z about 350 nanometer. And also come with a Earthscan fast mode. So instead of using the full detector, they're using the middle detector, using full same wavelength laser scan the field of view can achieve much faster speed. However, again, because the internal triangle, if you have a faster speed, then you're going to lose some resolution. So you can see the Y has a lower resolution compared with the X in this case. Just by comparison, for example, I was using a slide so-called ArgoSIM. This is a special manufacturer slide that has different patterns for different purposes. One of them is using a gradually increase this space every 30 nanometers. So in this case, from 0, 30, 60, and so on. So, and also by property manufacturer technology, this slide has never been bleached. So you can also use for quantitative analysis. So in this case, by comparing with the Widefield, you can see on the top, the top row here, and confocal with 1 every unit, and confocal with 0.25 every unit, and the image become much more noise. So there's less signal and more noise go to the detector. But however, the Airyscan can give you not only the intensity, but also the resolution, which is the best among all the situations. And here is the real example imaging the mitochondria. In the yellow, the zoomed in, you can see the confocal is kind of like a more noise, lower signal to noise ratio. But however, the Airyscan give you much higher signal to noise ratio, and also you can see some fine detail which is missing in the confocal image. In 2014, Nobel Prize in Chemistry given to the 3-person Eric Benzik, Stephen Howe, and then William Morner for the development of super resolved fluorescent microscopy. So at that time, even myself, I kind of curious why it's in chemistry, right? It's not in physics. I mean, optical should be counted as physics. So about the most important information here is the fluorescent microscopy. So even though we say we're talking about the super resolution today, all the technologies, but those are all based on fluorescent technologies or fluorescent microscopy. For the traditional one, for example, bright field, the DIC, those are still covered by the diffraction limit or Abbe's limitation, which is less than, 200 nanometer. So only fluorescent people using different tricks can go beyond the diffraction limit, less than 200 nanometer. So let's go over the most popular super resolution technique. So, and also, I was kind of curious about this person's name, Stephen Howe here, right? So by English, we all know the meaning of that. But actually, he's German, but he's actually Romanian. And but let's say just in German, if you go to the translation in German, it's in English, the meaning actually is bright or light. It's found out this kind of interesting. He won this prize. And also, if you use Zeiss microscope on the condenser, there's a filter wheel. On the condenser filter wheel, you're going to see a lighter H there. It's the same meaning for the Hell here. It's for the bright field in German. Okay. They didn't translate to English. They still use in German. And back to 2006, and Eric Benzik with his friend Harold Hess, and they saw an interesting protein called the photoactive fluorescent protein. And they have this idea, and then they contact the Jennifer Schwartz. And back then she was at the NIH. Now she's at the Geneva Research Campus right now. So they collaborate together, and they did some experiment called the photoactive localization microscopy, which is like Eric Benzik won Nobel Prize in chemistry in 2014. So the idea is this interesting property protein can be switched on and off by using different wavelengths laser. So in this case, for example, you can see from A, B, C, D, and so on. So by imaging this protein labeled sample, you can turn it on, and now you can bleach it, and then you can turn it on again. So you keep doing this many times, and because the point spur function is based on the Gaussian fitting, so you know exactly where to center the fluorescent protein, and then you know the exact coordinate. For example, become a spot here, and with time accumulation all those information, you're going to get a graph. So all this coordinate will generate a graph. So this paper was first published in Science back to 2006 September. And in the same year, another researcher, Xiaowei Zhang at Harvard, and her group did exactly the similar experiment, but instead of using photoactive protein, they're using a cyan dye. They use a Cy5, but the principle exactly the same. But she didn't win the Nobel Prize with them together. And instead, she won this breakthrough prize in life science back to 2019. But the idea is exactly the same. So the Eric Benzik using the fluorescent active protein, and Xiaowei Zhang group using this photoactive fluorescent dye. So because you know the point spur function, and by the Gaussian fitting, you know the coordinate. So by accumulated thousand images, then you get this so-called her group is called the STORM, Stochastic Optical Reconstruction Microscopy. It's just different names. And this paper was first published in Nature Medicine, actually earlier than Eric Benzik's paper. This was published in August. And Palm Storm, as I mentioned, they basically the same idea. So instead of using the conventional fluorescent microscopy, you turn on all the fluorophore at the same time. Therefore, like you turn on all the light on the sample, so everything will glow. So unless you use, like for example, confocal, eliminate the light out of focus, you can get a sharp image. Otherwise, you're going to have a blurry image. So the Palm Storm using a very clever idea, only randomly turn on fluorophore randomly on a sample, and by the Gaussian fitting, you know the coordinate. And with time, by collecting 1,000 images, finally you get this a graph here. So this actually is not an image; it's a graph, and the resolution is the highest among all the super resolution technologies. So the XY is about 20 nanometer, and then Z is about 50 nanometer. And just give you one example for the storm image. Here you can see on the left-hand side is the cell nucleus. And there you can also see the cell nucleus, nucleolus, which is a very dense protein structure inside the cell nucleus. And on the right-hand side, you can see the Widefield image. Usually when people doing colocalization image analysis, when they using two different labeling, red and green, and when they see yellow, they say colocalization. But if you use super resolution, for example, storm labeling two protein here, by the storm resolving, you can see actually they kind of like they don't look like a really colocalized. There's some fine structure detail in there, and because the storm has a very high resolution, which is kind of similar to the EM one, but the EM usually if you use the urine acid staining or the staining, it gives you very black dark image. You're not going to see any detail, but the storm can resolve some very fine detail. And Stephen Howe technology is called a stimulated emission depletion microscopy. And he first published this idea back to 1994. So the idea is when you use excitation light, bring the fluorophore go to excitation, when they go back to the ground state, will release the fluorescent. So by using a so-called STED laser, it's like a donut shape, by quenching off the outside of the fluorophore excitation, the emission. Therefore, you're going to get a much smaller signal here. So this is so-called a STED image. So for example, conventional confocal, you're going to see the light spot become like a huge pattern like this size. For X and Y, it's about 250. For the Z, it's about 500. But however, because it's a STED laser quenching off the outside the light, you only left the center light, therefore you achieve a much higher resolution. And here is one example, confocal with the STED. So with the technology advancement, so the current STED can resolve XY up to 30 nanometer and Z about 100 nanometer. Another super resolution technique, so-called structural illumination microscopy, and first introduced in year 2000 by GaAsP, and unfortunately he passed away in 2011. So therefore, he didn't win the Nobel Prize. I'm not sure if he will be counted as one of the Nobel Prize for considering for the super resolution technique or not. But unfortunately, he passed away. And him, together with John Sidat and David Agard, they first announced have this idea for structural illumination. This technology rather more difficult comparing with the previous, like the Palm Storm and also the STED. So by using a moiré pattern went through the sample and also make a rotation. So you're going to have like a different so-called phase and then frequency information. And those information will then later be transferred, become a Fourier transform. And with those frequency information, and we can reconstruct reconstruct the image with a much higher resolution. So basically, this is from the space frequency become a higher resolution. So the resolution for the X and Y can reach to 115 nanometer, and then Z is about 270 nanometer. So here's the two examples of the same images. So on the left, you can see the triple labeling for the BPAE cell. The left is showing the Widefield results, and then the right showing the CMIF results. You can see the CMIF give you a much sharper image. And on the right-hand side, it's showing you the Widefield and also the CMIF image for the actin. So if you see, I'm not sure, on my screen, I can see the resolution very well. You can see all those fine detail from the same image. It looks really amazing, and it's kind of like the some of the SEM structure. And at the CMIF, we acquired this Nikon so-called NSIM and InStorm super resolution microscope. And as I mentioned, we have the SIM resolution 115 nanometer in XY, 250 in Z, and Storm resolution 20 nanometer in X and Y, 50 in Z. And also come with a so-called TIRF, is total internal reflection fluorescent microscopy, which is used for the surface 200 nanometer. And this system also has the capability for the live cell imaging. So the TIRF phenomena are going to happen when light passes through from a high refract index media to a lower one. So for example, from glass to water, the glass has a high refract index about 1.5, the water about 1.3. And in certain angle, the light will be totally reflected back. So therefore, will form a very high resolution within the 200 nanometers. And the system also comes with the 6 lasers, with the standard 4 lasers, and also with the CIP and the YFP laser for fright applications. And the camera for SIM can go up to speed 15 frames per second. And the storm usually need to occur like a few thousand images. So usually the acquisition will be in minutes. So we have this instrument installed and fully in function. So if you're interested in, I can give you a tour, or if you have an experiment, you are welcome to contact us. We can provide the trainings on this sample, on these microscopes. Another very important term in life sciences is called the phototoxicity in microscopy. So the challenge for live imaging is the phototoxicity, because it can happen when you're using very intense laser shone on the sample, and the cell will release called the free radicals, which can damage the cells, very quickly, and also will stop some physiological conditions. So therefore, be gentle with the cell is very important. So and usually when you image for live cells, you want to use the longest excitation wavelength, which has the weakest energy, so based on the physics. And also it has the deeper penetration to the samples. So for example, in this time-lapse movie, you can see in the cyan, which is those are tubular structure, those are mitochondria. You can see with time, during the imaging, those mitochondria become dimmer and also become fragmented. So those because they are response to the phototoxicity to the lasers. Okay, so therefore, when you do the live cell imaging, you should use a much lower laser power, much lower illumination power. But the problem is if you use lower illumination power, then you're going to end up a poor signal to noise ratio. So in recent years, the artificial intelligence become more and more popular. So, and also the deep learning. So, but the deep learning actually is only a small part of the so-called AI is part of also smart part of the machine learning. And we heard this name more and more popular in many applications, especially in the autopilot in cars right now. So basically, you can provide lots of training data to the to so-called neural network, and after then, it gives you much better output magically. So for example, we have a Nikon Confocal come with a so-called Denoise.ai, which is a pre-trained neural network. So the life for especially useful for resonance scan, instead of like confocal point scanning, the resonance scan speed is much faster than point scanning. But the problem again, if you reach to a faster speed, you're going to end up with a poor signal to noise ratio. So for example, on the left-hand side, you can see this picture looks very noisy because using the resonance scan, you can have a faster speed. However, the resolution and the signal to noise ratio become lower, the sensitivity become lower. With the Denoise.ai pre-trained neural network, by removing from the detector the short noise, you can get a much sharper image and with a much higher intensity for all the color. You can see the nucleus, mitochondria, and also the actin. This is one application. I believe the same thing you have application for the EM imagings, and also yeah, for EM microscopy and deep imaging. Due to the light scattering, the image depth usually in regular tissues for the 1 photon, which for example like confocal cannot go beyond more than 200 micrometers, and for 2 photon or multi-photon usually limited by about 1 millimeter or about 800 micrometer. And back to 1931, as Maria Gapter-Mayer, she first have this proposed this idea for 2-photon excitation. Is it possible in her PhD thesis? So instead of using one high energy laser, pop the fluorophore to excitation state, you can use a much weaker energy using a longer wavelength, using do it twice, pop this fluorophore to excitation, if you achieve the same results. But this idea couldn't be tested until 1961. As I mentioned before, the laser was first invented in 1960. So before then, this idea couldn't be tested because the light source is not powerful enough. And then in 1961, Wolfgang Kaiser, he tested this 2-photon idea that is possible. And again, until 1990s, and by Winfried Dank, and with his colleagues at the Cornell University, by then he was a postdoc. So they were able to successfully develop this commercialized 2-photon microscope. So for example, in this picture, you can see the single photon, the light, when the light, here's the light focus, but beyond and below the focus, there are also extra light. Those light can also make the image much more blurry, go to the detector, and also there are more photo damaging to the samples. And however, for the 2-photon or multi-photon, you can see the light only focus on this spot here. I'm not sure you can see it here. It's a very tiny spot here. So therefore, they achieved the optical sectioning by this 2-photon excitation. And also because it's so precise focus on just have this on the focus. So you can also use the, and also this because the laser is powerful enough, you can also use this laser for for precise injury damage to the samples. And here's just one example imaging. Usually people use this for in neuroscience imaging the mouse brain, for example, in live animals, and they can image much deeper into the tissue, for example. And in 2013, Carl Desro's lab at Stanford University, they pioneered this technology called the Clarity. But this technology only applies to fixed samples. So, but the multi-photon or two-photon can apply to live animals, for example. So they cleverly found out that by chemically removing the lipids in the tissue and make the tissue become transparent, after matching the refract index, the tissue will totally become transparent. So by then, the technology is everything's minute and takes very long time. For example, from the beginning to the end, it takes about 9 or 10 days to get the results. And by the way, he's also the person who invented so-called optical genetics, which is widely used in neuroscience. It become very powerful tool to study neuroscience. And also he won the breakthrough prize for the optical genetics in 2016. And after then, there are many different labs developing these clearing techniques. And there are many different clearing techniques right now. This is, for example, there's some very popular one, and these are different refract index here. For example, the one from Desro's lab is using the hydrogel, which is refract index about 1.45. And a company, Logos Biosystems, they make this technology become much more easier. So they commercialize this this technology and instrument, which is very easy to use, a so-called X Clarity. So just within 15 hours, you can transform this mouse brain with a spinal cord become much clearer tissue. This is kind of like open a window on a black box, right? Before the brain looks like this, because the light scattering, you cannot see very deep. Even with the two-photon is limited less than 1 millimeter. But with this technology, everything becomes transparent. You can see much deeper. And the reason you still can see this entire tissue here is because this tissue is in air. The air has different refract index than the tissue. Therefore, you can see it. But after matching the refract index of 1.45, this tissue will become transparent, almost invisible in the solution. So before I show you the results, I want to also talk a little bit more about the so-called light sheet, or called the single plane illumination microscopy. The idea was introduced by a long time ago in 1902 by Richard Zsigmondy. So back then he invented a technology so-called ultra microscopy by using a sheet of light goes through as a light source and then can achieve much higher resolution. So later developed a so-called light sheet. So instead of like a confocal, using a pinhole, so the light sheet using a sheet of light as an optical section go through this tissue. For example, in this here, the light and then red is the objective used as an illumination objective and illumination source. You can see this light will become like a sheet here, and usually it is as a laser. And there's another detection objective which is 90-degree perpendicular to this light sheet, the sheet of light. Therefore, you can only see the sheet of light sample become fluorescent and in focus. And also because the sheet of light is much more gentle compared with all the illumination to the entire sample, therefore there is much less phototoxicity to the sample. So that also means less photobleaching. So you can see if the idea for the regular epifluorescent illumination, the entire sample will be shone up by the light, but the light sheet only use a thin layer as a light source to shine on the sample. But as you can see, because the light diffraction, the light sheet certainly work better with a much transparent sample, which is widely used for developing biology. For example, in zebrafish or Drosophila flies or some plants. But with the clearing technology, tissues can also be used for this technology. So for example, on the left is showing you the lectin perfuse the mouse brain blood vessels. So the light sheet can image much deeper. In this case, 2.3 millimeter. However, the confocal can only image less than 1 millimeter by depth, and also takes much longer time. And also, they are much more photobleaching due to the phototoxicity to the sample. You can see the light sheet, the fine detailed structure, but however, the confocal, the entire structure been destroyed. And also by the way you put the sample in the instrument, the light sheet can image the sample even with a 360 rotation, but the confocal usually put the sample on the stage, which is only X and Y. Due to the distance, if you image deeper and deeper, the sample actually further away from the illumination source and become dimmer. And also the detector become dimmer. So you can't see very well. So here is one example. It's only a small part of the mouse brain. The red label for the blood vessel, and the green is a virus injected label the neurons. And in this case, you can easily quantitative analyze the injected neuron by location, connection, and also count the numbers. So this also treated by the software using the surface rendering, so you can see the structure much easier. Okay, so that's all for my talks today, and thanks for your attention.